Welcome to the Ashtakar Frontiers of Science lectures in the Eberly College of Science. Good morning, I'm Charles Anderson, Associate Professor of Biology in the Eberly College of Science and the Chair of our college's Sustainability Council. I'll be your moderator for this year's lecture series. Before we introduce our speaker, I wanted to go over a few housekeeping notes. We will be recording today's lecture, which will be posted on the lecture series website as soon as it is ready. At the end of the lecture, there will be a question and answer session. You can use the Q&A function at the bottom of your screen to enter your questions, which I will share with our speaker. The lecture series was founded by Abhay Ashtakar, founding director of the Institute for Gravitation and the Cosmos and a member of the National Academy of Sciences. It owes its success to Barbara Kennedy, who presided over the series during its first 25 years, making it one of the most successful science outreach events in Pennsylvania. This year, the Ashtakar Frontiers of Science lectures are entitled Sustainability, How Science Can Help Achieve the UN's 2030 Sustainable Development Goals. The series will focus on the United Nations 17 Sustainable Development Goals, or SDGs, which are a call to action to end poverty, protect the planet, and ensure that all people enjoy peace and prosperity by 2030. Today's featured speaker is Daniel Cosgrove, a professor in the Department of Biology at Penn State. Dr. Cosgrove studies how plant cells grow, with a focus on the structure of extensible plant cell walls and the mechanisms by which they stretch irreversibly during growth. He is the founding director of the Center for Lignocellulose Structure and Formation, an Energy Frontiers research center funded by the U.S. Department of Energy since 2009 that operates as a coordinated effort by 16 research groups at 7 universities and Oak Ridge National Laboratory. The center's mission is to develop a nanoscale understanding of cellulosic cell walls, the energy-rich structural material in plants, forming the foundation for new technologies in sustainable energy and novel biomaterials. This effort is relevant to SDG 7, affordable and clean energy. Following his undergraduate and graduate studies at the University of Massachusetts and Stanford University, respectively, Dr. Cosgrove went to Germany and Seattle for research visits before joining the biology department at Penn State, back, as he puts it, when dinosaurs still roamed Happy Valley. Today, Dr. Cosgrove will deliver a presentation based upon his research, entitled Sunshine into Wood, How Plants Build Their Cell Walls, a Gigaton Source of Renewable Materials and Energy. Dr. Cosgrove, thank you so much for being here today. We look forward to learning more about this very interesting and relevant topic.
Thank you, Charlie. Thank you, everyone, for tuning in on this cold February day. Happy Chinese New Year, Valentine's Day weekend, and Carnival. The convergence of multiple traditional holidays, which are being celebrated very differently this year in the year of the COVID. So what I'd like to tell you about is some of the research from my lab, but also primarily that focused in this center that Charlie mentioned. And let's just jump into right into it to talk about plants and plant cell walls. So those of you who have been to California probably visited Yosemite National Park and have met some of these wonderful creatures, massive organisms, single plants of massive weight. All of this starts with a very tiny seed, and so in the course of their lifetime, they manage to collect CO2 and water, and through the magic of photosynthesis, build this into these massive bodies. I visited this a few years ago with my family. Here's me and some of them under one of these trees in the shadow of a hauled out tree. Terrible thing to do. And here I am with my nephew resting against one of those, the tree trunks. Now, in Pennsylvania, we don't have redwoods, and the trees are more diminutive in size. This is a stump that I photographed from local Black Moshannon State Park, and it provides some information about the structure of these of this material, this body of the plant. And so this was taken just with my phone camera, and just with that limited technology, one can see some of the organizational properties of woody materials, and even some aspects of cellular organization, or at least the way the cells are lined up relative to one another. If you want to get more details of cell walls, you have to use some more specialized techniques. So here are some thin sections viewed with electron microscopy, and another type of image of the cell wall, giving you a taste of what this material looks like at the micrometer level, at the cellular level. And wood anatomists have categorized wood structure, and they stain these materials with metachromatic dyes, in which you get information about the different kinds of materials that are in this cross section of wood. But also, I find these slides very pretty. They're just as an image of abstract art. They're very attractive objects. If you want more details about the cell wall, though, you've got to go even finer details. So here's an electron micrograph of a cross section of a cell wall, and it's split, and you can see some of the fibrillar nature of the cell wall, where pointed to by the arrow. That still provides relatively limited information, and so to get more information, you need to apply different kinds of techniques, and I'll be telling you a little bit about some of our approaches for that. This is an image or an artistic representation, an artistic rendition of woody cell wall material, showing some of the components. So the green object here is a macrofibril of cellulose, and we'll have more to say about that, but you can see it has a hierarchical organization. The brown masses of material are representations of lignin, and those are the major objects in wood, so hence lignocellulose. And then there are other materials in there, matrix polysaccharides, matrix polymers of various sorts that I won't need to go into at this scale. But this is what one imagines the cell wall being organized at the almost molecular level. Now, globally, there's a massive amount of this material that's made. So plants, of course, collect solar energy and CO2 from the atmosphere, combine that with a bit of water to make this material. Some 200 billion tons of biomass are annually produced by land plants in the year, according to some estimates. Most of that ends up in plant cell walls. 1% of that is used by human society for materials, so paper and lumber. 1% is used for energy in the form of cooking and electricity generation. And 98% of it is used by microorganisms to fuel their lifestyle. So this material is ultimately broken down by bacteria and fungi that eat this stuff for breakfast. So 160 billion tons is a little bit hard to fathom. I made a quick calculation. So probably in central Pennsylvania, you're familiar with a cord of firewood. I heated my house for years with firewood, and I moved and stacked firewood. A cord of firewood is about 4 feet by 4 feet by 8 feet of stacked wood, and that weighs about 1 ton if it's properly seasoned. If you get wet firewood, it doesn't burn so well, and it'll be a lot heavier. So it'll heat you up as you stack it, but it won't provide so much heat for your home. So 1 billion tons of wood could be compared with a line of cords packed end to end from New York City to Seattle in my calculation. So 160 billion tons of cord means you would stack that line that runs basically along the border from Canada to the US, in case the Canadians get some ideas about keeping Americans from joining their country. 160 cords would amount to a wooden wall 600 feet high or so. I've driven from New York to Seattle multiple times, and it's a long, tedious drive, and maybe this helps you represent how much material is being produced annually as cell walls. So plants are used in many material forms by human society. So of course, you know, lumber. You're probably aware that forages are primarily cell walls, and agriculture depends upon that heavily. Fibers, cotton, ramie, and other textiles are essentially pure cellulose. Paper is also almost pure cellulose, and that all comes from plant cell walls. There's a number of polymers that are used in films and plastics. That use has fallen off since the 1960s as a result of plastics coming from the petroleum industry, undercutting the price of cellulosic materials. But this is seeing a reversal recently, where a lot of companies are interested in reverting back to cellulose-based polymers for plastics because they're biodegradable, and so they don't have a long lifetime in the environment. Organisms know how to break this stuff down. So a lot of interest in plant cell wall derived materials. Recently, the technology of wood has been changing, and so there are ways of making much stronger wood materials, laminating them, etc., such that they can replace steel, and there's many efforts afoot to replace steel in high-rise buildings. Paper last year talked about a procedure for condensing cell walls into something that is very strong. It's what's called superwood. Take a look at it. A lot of interesting articles on that on the web. And another publication also from another group in Maryland, where the superwood originated, shows ways of making wood with different properties, modifying wood so it has different properties. Okay, let's see. I can see I can't start that movie without the laser pointer. Okay, there we go. All right. So this is woody material has been slightly modified, so it becomes highly squishy and elastic. And here's a version of it where its elasticity, its recoverable energy is shown. You can make super balls out of the woody material. Normally, if you tried to do this with regular wood, it would just thump right away. It's a poor super ball. It doesn't have the energy recovery of the best super balls, but it's still much different from natural wood. There's also interested in cellulosics, nanocellulosics as a material for making new kinds of materials, and that's been something that's been burgeoning in the last 20 years and is really taking off. Cell walls are also an important energy source. If you sat around a fire, you're quite aware of that's the case. Some of you familiar with Greek mythology know that the Greeks have a myth about Prometheus bringing fire to human beings, and for that he engendered the anger of Zeus, who rewarded him by chaining him to a rock and ordering his pet eagle to pick out his liver every day. Since Prometheus was immortal, his liver would regenerate overnight, and he was chained to suffer that over and over again until somebody rescued him. Forest fires represent in an impressive way the energy stored in wood, and of course in California we see the devastating aspects of that. There's also, for the last 20 years, been intense interest in some circles to generate biofuels from cellulosic materials for the transportation industry. That has its pluses and its minuses. I won't go into those arguments at the moment. But there's also efforts to convert woody materials into other kinds of biochemicals for industrial uses. So let's go back to plants. That's my love. So plants form cell walls, and a lot of the work we've been doing recently is centered in this Energy Frontier Research Center, shown here. And let me just say a couple of words about it. The focus of this center, which is funded by the Department of Energy, is to understand how plant cell walls make how plant cells make the cell walls and what the cell walls are, how they're structured. And so that begins with the machinery that synthesizes cellulose. It also focuses on the structure of cellulose and then how cellulose interacts with other components to make the real cell wall. This material all starts from photosynthesis with a byproduct of photosynthesis, major product of photosynthesis, glucose, and the cellulose synthesis machinery strings glucose chains together to make cellulose. Now glucose is a nice low molecular weight soluble material. We use it in our bodies for energy. Cellulose, on the other hand, has very different properties, and that's a consequence of the physics of polymerization of these glucose polymers. Cells also then make other materials, and all of this gets assembled into this complex structure that I talked about. So just as a quick aside, glucose is an amazing molecule. Plants can use it to make cellulose by certain bonding arrangements. They can also use starch, same material, but just bonded in a slightly different way. And this has very different physical properties. And of course, starch is easily digestible by humans, whereas cellulose is not. In humans, we store glycogen, which is a glucose polymer that's chemically related to starch. Glucose is a key metabolic energy for all living cells. It's also blood sugar, right? And if you have bad control of blood sugar, too high a concentration of it, that's diabetes, and that will lead to some detrimental consequences for your body. It's interesting to consider that some plant tissues can suffer very high concentrations of glucose, apparently without the high detrimental effects of glucose. And how plants do this, I'm not sure is understood. All right, so back to cellulose and back to our plant cell walls. So this center, as Charlie mentioned at the beginning, is to understand the structure and the formation of the cell wall. And what we do is we combine the tools of physics and biology to understand this. So what's the nanomachinery that converts simple sugars into things like cellulose microfibrils? And then what are the physical processes by which cellulose interacts with other components to make something that has very distinctive and versatile and dynamic properties, the cell wall. I should mention that this center is led by Penn State, but it includes 6 other institutions, and they're important for how we operate. So this center is focused around 4 major skill sets. So biology and structural biology, imaging, polymer physics, and computational chemistry. And we use the techniques that are used in these different fields. So that involves things like mutagenesis, genetics, recombinant DNA, CRISPR, technologies that you no doubt heard about in the news, and also structural biology tools such as cryo-electron microscopy and protein crystallography, a wide variety of microscopies, including some fairly expensive instruments that require central facilities to use them, as well as spectroscopies, likewise, that require some very expensive instruments, nationwide instruments that are supported by the Department of Energy. And then there's a good dose of other physical techniques such as nuclear magnetic resonance and wall mechanics. And then all of this is tied together with various modeling approaches. This is the scheme that we are following for our research. So we begin with Well, we begin in the middle actually with cellulose. That's the hero of our story. It's and its properties, and I'll have a little bit more to say about that. Our center is also focusing on how cellulose is made, so that involves the enzymes and the complexes of enzymes that make this cellulosic material. And then lastly, how this integrates into the cell wall. So let's look at this cell wall. This is an image from my lab of a surface of an onion epidermis. And you can see very beautifully the individual cellulose microfibrils and how they're organized into layers. It's a polylamellate structure. And pulling together other information from the literature, we know that this microfibril consists of individual strands of a beta-1,4-glucan. So that's glucose chains hooked together end to end. But that by itself doesn't make cellulose. Cellulose is an array of these that are organized into a crystalline order, as shown on the right-hand image here. One representation of the crystalline order of cellulose. So this is, we believe there's 18 chains in highly crystalline order in this microfibril. The dimensions of this is about 3 nanometers, and by comparison, the Watson and Crick DNA structure is about 2 nanometers in diameter. So this is pretty small stuff. And yet it has some remarkable properties, very different from DNA. So DNA, for example, in our body, they say there's about a meter of DNA that is packed into each of our nuclei. And that is not possible if you took a meter of cellulose and tried to pack it into something as small as a nucleus, because it's a very stiff object. It has a very high, not only a small diameter, but a very long length. And so that gives it some high aspect ratio that has attractive features for material science. It's stiff and it's rigid. The crystalline structure of cellulose means that it has different physical properties on different facets of this crystal. And that's important for how the cell wall is constructed. And it's also important for how it's used in material science, for example. And cellulose is very resistant to enzymatic attack. So that's why it doesn't get digested in your stomachs. It's the individual chains are easily digested by enzymes, but when they're packed into this crystalline array, they exclude water, and enzymes have a difficult time separating out a single chain in order to get a bite on this oxygen bonded interface between individual glucose chains. And as a consequence, it's very challenging to break it down with enzymes. How is this made? So here's one representation of the structure that plants use to make this cellulose microfibril. It's made by a multimeric complex, a supramolecular organization. I'll have a bit more to say about that in a second. That's embedded in the surface membrane of the cell, the plasma membrane, and it takes glucose units from the inside of the cell and strings them together into a polymer that gets transported across or through the membrane. There's the protein forms a tunnel that allows the chain to get out, and then 18 of those chains are organized to form a crystalline microfibril. Now that's pretty magical. Human technology can't make anything with this degree of order and structural strength and at this dimension. So plants are doing things that rival the best human technologies. This complex is not static. It moves through the membrane, and it's guided by some of the cellular machinery inside the cell. And as a consequence, that organizes the way the cellulose microfibrils relate to one another, and that in turn has a big effect on the mechanical properties of that cell wall. So how do we know about this? That picture that I just showed was a convergence of 3 very different approaches taken by different groups in the Center for Lignocellulose Structure and Formation. So one approach is based upon what's called freeze fracture electron microscopy. So if you prepare a cell properly and you freeze it and then crack it in a certain way, there'll be a fracture that'll go through the membranes of the cells. And this can then be imaged with electron microscopy. And what you can see are objects shown at the top here. There's a rosette-like structure, a hexameric structure that is the synthase that makes the cellulose microfibril. And there's been some discussion about what's in each of these little particles. So that's what you can resolve with electron microscopy. You can also take the genes for the cellulose synthase enzymes and express them, for example, by getting bacteria to make these proteins, secrete them out into solution, and then use technologies such as X-ray scattering and neutron scattering to learn something about the overall shape and organization of these enzymes. And combining that with computer modeling of the enzyme structure along with the electron microscopy data, one can then try to fit all the pieces together to figure out how many of those synthases are in this complex. And the picture that emerges from this is shown here on the right. So this conclusion is shown here, this hexameric structure. Each of those particles has room for 3 individual cellulose synthases, 3 different enzymes, packed in a regular order. Now, just last year, a research group in Virginia, as part of our CLSF, part of our center, published a cryo-electron microscopy-based structure of the cellulose synthase found in a trimeric complex. So that's those individual particles in the hexameric particle structure. And this is the image that shows up. So 3 cellulose synthases, they're shown here as SAs. They're each colored with a different color. They combine to make a trimeric complex. And you can see the view looking from the cytoplasmic side on the left here with this stalk structure that extends into the cytoplasm. Or from the side, you can see the large globular top part of it that's within the membrane of the cell. So this structure then gives us a lot of additional information about this machinery. Just recently, in fact, this month, this structure was designated as the PDB molecule of the month for February 2021. Structural biologists are like other humans. They like to point out cool things to the public, and this evidently is viewed by the PDB people. This is a protein database, a large central for structural proteins. Like this, and a molecular artist by the name of David Goodsell, who's associated with the PDB organization, put together this watercolor of the cellulose synthase complex. And you can see here each trimer with 3 cellulose synthases, shown here in slightly different colors of green. And then each of these trimers is packed into a hexameric organization, making this circular structure. And you can see from the original cryo-EM structure, you can see glucan chains emerging from each of these synthases. And so there's a total of 18 of these emerging from the surface. We're looking down now from the outer side of a cell, for example, at what would be emerging, what would be the cell wall side of things. And these glucan chains then coalesce in some way that we still don't understand to make this highly crystalline microfibril. The same artist made an artistic rendition of the cell wall. And what you can see here are the cellulose synthases. There's 2 of them shown here, these complexes in the membrane of the cell. That's shown here in light purple. Down at the bottom in blue is the cytoplasm packed with various structures. And on the outside in the the brownish tones is the cell wall. And you have to imagine that these cellulose synthase complexes are not static, but in fact they are moving through the membrane, perhaps being pushed by the crystallization of the individual glucan chains into this crystalline microfibril, and that drives or is thought to provide a motive force for pushing this complex through the membrane, and as it does so, it leaves behind a trail representing the microfibril. So why do we care about this? 2 points I want to make. So understanding the molecular processes by which plants make cellulose from glucose units is a key goal of our center, and it's a fundamental principle of science that you need to understand how the universe works, and that can then be used by engineers to then design and modify the original materials for into new processes and new materials. So this serves as a basis for engineering this nanomachine using genetic techniques, for example, to make cellulose with novel properties. So it might be possible in the future to modify the chain length of the cellulose polymers, the crystallinity of the cellulose, the microfibril length, its surface properties, and all of these are key physical properties that make wood what it is, make paper what it is, make cotton and what it is, and these other polymer-based plastic type materials, what they are. So being able to understand how the material is synthesized, we're in a better position to use that knowledge to engineer this for new purposes. So what I've talked about so far has been the left-hand part of this. I want to now focus on the cell wall, and this gets much closer to the heart of my research. And the topic I'll talk about is in this part is our attempt to relate mechanics, so biomechanics, to cell wall structure and to the growth of the cell. And so there's three steps in this process. One is understanding the structure of the growing cell wall, and we use a technique called atomic force microscopy for that. A second technique is making mechanical measurements of the cell wall, and then the third one is to try to bring this information together through what's called a coarse grained model of cell wall structure. So just a reminder, plants get really big, and that's a consequence of cell enlargement. If cells grew the way your liver cell grew, the tallest tree in the world would stand less than waist high. That's a consequence of massive cell enlargement that occurs by plant cells, and that's something that's more or less peculiar to plants. And my lab's very interested in understanding how that is controlled, how that is regulated. And that brings us down to what's the structure of this highly extensible cell wall, and what are the physical and molecular mechanisms controlling cell wall enlargement. So one of the systems we're using for understanding the structure of the cell wall and investigating its mechanics is something that you all have experience with. So this starts with onions. And onions, it turns out, have a property that you can peel the epidermis. And when you peel that epidermis, you get a single layer of cells. And it's not really a single layer of cells, it's a single layer of just the outer epidermal cell walls. So the cells are all removed, and all you're left with is a single layer of cell walls that makes a nice continuous sheet. That cell wall layer is about 7 micrometers in thickness. So it's a very thin object. And we can take that material, put it under a atomic force microscope and image it and carry out nanomechanical measurements of the cell wall. We can also take this large sheet of cell walls and stretch it and make other measurements to assess the mechanical properties of the cell wall. So this is what we see when we look at this wall in the hydrated state with the AFM, 2 different scales. And you can see fibrillar organization, and it's a polylamellar organization. There are multiple lamellae. The cellulose microfibrils are not individual microfibrils. In fact, they show extensive bundling and network formation. we can stretch the cell wall under the AFM, and with a bit of finesse, we can see how the microfibrils are changing their organization, their structure, how they change within the cell wall as we're stretching it elastically, and that's shown in this image on the left. Or we can also make the wall undergo long-term extension, and so that's what we call creep, and study how the cellulose microfibrils change in that condition. And it takes some time to study these things to see what's going on. The point is that the behaviors of the cell wall microfibrils are remarkably different under elastic extension, so elastic reversible extension, versus the kind of long-term irreversible extension that occurs during growth, for example. The other thing we can do with the atomic force microscope, we can not only get images. The microscope works a bit like a stylus on the old-fashioned record player. It rides up and down on the surface, and it scans across the object. You get a topology, a height map that's shown here in the red. But at each point, it can press down into that surface and measure the resistance of the surface to the force used to press down on it. And so from this, you can get a stiffness or a modulus. You can get something, other properties such as the deformation of the object, and you can also get adhesion, how sticky is that material to the tip. And by combining these different maps, these different nanomechanical maps with the height map, we can learn some things. So for example, we can see regions where the microfibrils are supported. They don't deform as much to the indentation with the AFM. There's other regions where the microfibrils deform quite a lot. So the underlying matrix for them is not supporting them. We can also see non-fibrillar regions. So this is not cellulose that is here, but it's some matrix component that evidently is fairly stiff. We can see regions with high adhesion. So we think of these as pectins. So pectins form the matrix. Pectins are acidic polysaccharides in the cell wall, and they're the same things that cause a gelling of pies, for example, or jellies. And so we move from that to larger scale mechanical studies, where we can stretch the cell wall in various ways and assess its properties. Some of these are shown here. So on the right, we can stretch the cell wall and ask, how does it behave with long-term irreversible extension of the cell wall? We can also stretch the cell wall in the green panel here very quickly and reversibly and get its elastic and plastic properties. We can indent the cell wall with the AFM, so poking a needle from the side, and get some mechanical properties at right angles to the tensile or in-plane direction of the cell wall. And then with the AFM, we can see how the microfibrils move as we subject them to a dragging force. So we can measure the lateral motion of microfibrils. I'll just show you one movie of this mechanical assay. Here I've got the onion cell wall epidermis between two clamps. It's a 5mm stretch, and you can see this transparent object here is the cellulose microfibril, or excuse me, is the onion epidermis. You can see cellulose. You can see cells. If you look closely, there's just glimmers of the cellulose structure of this material. And we stretch it once, and then we return it and we stretch it a second time. And from this, we can get force extension curves or stress strain curves. We can assess the mechanical properties and get plastic and elastic properties of the cell wall. And so from these kinds of and related studies, we can learn that the microfibrils in the cell wall are the dominant controlling factors for the tensile responses, so stretching in plane of the cell wall. Pectins have a very modest effect. From the indentation, however, pectins are important. They're influencing how the wall responds to indentation behaviors. And there is a smaller effect of microfibrils. Now we can take this mechanical behavior and try to integrate it with our view of cell wall structure. And here, a slight diversion. This is something as director of the center, I quickly learned that physicists and biologists think of models in very different ways. So when a biologist thinks of a model and presents a model, this is the kind of thing that person is thinking about. So you see cellular objects, you see where they're organized in space and how they contact one another, and maybe there's a little bit of dynamics involved in that. So to take this to an extreme case, you could also say this is a model of as represented by biologists, and maybe it's not so far-fetched. On the other hand, when a physicist thinks about models, this is what they think about, equations. And so it's a very specific quantitative relationship expressing one thing to another thing in explicit terms. Each of these kinds of models have their advantages and disadvantages. It took us a little while to learn that when the biologists talk about models and the physicists talk about models, we're not talking the same language. So we've recently teamed up with an engineer. My group has teamed up with an engineer, Professor Su-Lin Zhang in the Engineering and Science Mechanics department here at Penn State, to develop a model, a physical model, based upon the structural model from what we see with electron microscopy, atomic force microscopy, and other sorts of things of the cell wall. And this is to tie together mechanics and structure. So the approach is called coarse grain modeling, and what you do is you represent a polymer as a string of beads. They're hooked together by springs, so this represents the stiffness of the polymer, resistance to stretching. There's also some other parameters of the model, so these are all very physical representations. And you can also represent the lateral non-covalent interactions, if you will, the stickiness, the adhesion, the binding of polymers to one another. And in our case, we represented the wall with three major constituents that have very different properties. So cellulose, pectin that I mentioned, this acidic polysaccharide, and then another polysaccharide that's in primary cell walls called xyloglucan. And so we were asking these kinds of questions. So can simple physical interactions lead to microfibril networks that resemble the cell wall? What kinds of interactions between polysaccharides result in the mechanical responses we see in real walls? And what kinds of insights can we generate on the structural basis for wall assembly mechanics and chemorheology? So our model, in short, looks something like this. We represent our cell wall as with three lamellae, organizations of these three components, polymer components, and here's some images of that. The four lamellae have a different cellulose organization in each case, representing what the onion cell wall, epidermal cell wall looks like. And then we take this and ask some questions. So how does the model respond to stress strain experiments, and how does that compare with the real wall? So I already showed you a force extension or a stress strain experiment with the onion cell wall, and some of those data are shown in this graph on the dotted lines, and then the model is shown in solid blue, and you can see the two curves overlap very well. So the model recapitulates tensile mechanics very well. The other thing we can do is then start asking more molecular questions. So how is the stress in the cell wall, how is it distributed among the different components of the cell wall, cellulose, pectin, xyloglucan? How is it distributed in the various lamellae of the cell wall? And this figure on the right shows a heat map with the color coding for the stress in the cellulose microfibrils in four lamellae, which are organized in different directions after we've stretched the cell wall in the horizontal direction. And what you can see is the lamellae have different colors, indicating they are bearing different loads. So it's not a nice uniform load-bearing structure. It is has very selective properties for load bearing. We can also image how the wall model stretches and see dynamically how it's behaving. So if you follow where some of these arrows are, you can see, for example, microfibrils changing their organization. They undergo bundling and unbundling. They undergo sliding. And all of this is important in dynamically changing the structure of the cell wall as the cell wall is stretching. We can also look closely and see how microfibrils change, how they show different kinds of movements. So they show, for example, sliding. We think this is an important aspect of the mechanics of the cell wall. They undergo straightening, they undergo curving, they undergo different kinds of bundling, they undergo reorientation. So these complex behaviors are all part and parcel of how the cell wall behaves as you stretch it. And so we're getting a molecular interpretation and an understanding of the macroscopic stress-strain behaviors of the cell wall. So why do we care about this? Well, it provides a means to understand and predict how changes in cell wall mechanics emerge from the molecular structure of the cell wall. And this provides a basis for engineering cell walls with novel physical properties, novel mechanical properties. And this is important for material science and agriculture and related fields. Agriculture, because these cell walls are parts of plants that control the growth of the whole plant. All right, so what I've said is these plant cell walls are sophisticated structures, and they have quite versatile properties. Think of the physical properties of a chunk of lignocellulose, a chunk of wood versus a piece of onion skin. They are synthesized by plants from very simple sunlight, water, and CO2 using sophisticated processes and sophisticated biological devices. This is important to society in the form of renewable materials, energy, and also as a global scale carbon sink, which is of course important for countering the increasing rise of CO2 in the atmosphere. And I also can't fail to emphasize that they're also important to plants as well. These things control the growth and morphology and structure and mechanics of plants. So trees would not be trees without their cell walls. They would be blobs of amoebae, just slime. Okay, so let me wrap up with a couple of quick acknowledgements. Who we are in CLSF. Here are some of the faces for the senior investigators. Each of these is leading a group at their own institution. And I want to give thanks to all of these. I've only had an opportunity to represent a slice of the research, a small slice of the research that's occurring in our center. All of us are contributing complementary and important skills to understanding the cell wall structure. And for people that I interact directly, here are some images of some of the key people here with their names, students, graduate students, technicians, postdocs, and management staff. So thank you all to them, and also thank you to the Department of Energy for funding this work. And the last thing I want to leave you with is my artistic representation of a plant cell wall. It starts with an electron micrograph, or excuse me, an atomic force micrograph shown here on the left. A higher scale image of that is shown on the right, and where I've used a pen to colorize the individual microfibrils at the surface. And then using a variety of other information that we have, I color in what we think is the likely organization of the two major other components in this primary cell wall, that is the pectins, these soft acidic polysaccharides shown in yellow, and the xyloglucans, which are polysaccharides that have a strong affinity for cellulose surfaces. And I will stop there and will be happy to take questions.
Dr. Cosgrove, thank you so much for sharing your inspiring work with us, and I applaud you on behalf of the audience. We will now open the floor for 30 minutes of questions from our audience. And I'll start out, and full disclosure, I am a member of the center, so I know a little bit about this research. But I'm curious, Dr. Cosgrove, to get your perspective. What do you think it's like doing research as part of a larger multi-institute, multi-investigator center in contrast to doing research as a single lab? And before you answer, I'll encourage people to, again, put their questions in the Q&A box at the bottom, and I will read those out to Dr. Cosgrove.
So I have experience in both work styles. So I've been working on cell walls for many years, and in the first phase I was using the typical mode, which is a single PI, head of a lab, getting individual grants on specific projects, and occasionally I would get collaborators to help out with certain aspect of it. But that's the mode for much of academic research. The Department of Energy started these projects, these Energy Frontiers Research Centers, 12 years ago with a very different model. So their idea was to put together teams of experts with very complementary specialties to tackle major problems that are not feasible to tackle in a single PI environment. And that's how we designed it. I must say that for me, it's been a great experience interacting with physicists and chemists and others. And I've learned a tremendous amount as a consequence of that. I've learned a tremendous amount about how to interact with researchers from various different fields, but also I've learned a tremendous amount about other aspects of plant cell walls and other kinds of approaches to tackling cell walls. So we take very different approaches, and we're learning that. I think we're also having our early career researchers, so the graduate students, undergraduates, postdocs, participate in this environment, usually in teams, working together with others with different expertise. And the feedback I get is that they find it a gratifying experience. We have multiple monthly meetings where we're reporting various aspects of it, and I believe and hope that they find this a stimulating environment. So I've drunk the Kool-Aid, as we said, from someone from who grew up in the 1960s. I think this approach works. The centers can be too big and then they don't interact and they're not effective, but I think the size of this sort of a center with a dozen or so groups interacting with complementary expertise really provides a very different experience for pushing the science forward and also for educating and training the new generation of scientists.
Great, thank you. One of our audience members asks, how do the properties of the cell wall change as trees are processed into lumber?
You know, I'll answer as a layperson, right? I don't have a experience in forestry or lumber making, etc. But the way I understand they're being processed is the wood is cut and kiln dried. Typically, that's all that's done, I believe, before it goes out to market. Sometimes it's embedded with chemicals to reduce fungal growth, right? Toxic chemicals, and so that's an additional process. So what happens is water is driven out of the material, and when you remove water from something that's polysaccharide based, you change its properties substantially, its physical properties substantially. And of course, you all know this from general experience. If you take a polysaccharide, you take starch in the form of pasta, spaghetti, right? Out of the box, it's stiff, rigid, brittle, has very different physical properties from after you've hydrated it by boiling it in water for a couple of minutes. And that boiling in water makes it much more flexible, stretchy, elastic, and a tougher material. It's not nearly as brittle. So it changes its physical properties quite a lot. It increases in strength. Wet wood tends to be deformable. If you put it under constant pressure, it will slowly creep. As a building material, that's usually not so good. And so the wood engineers are devising ways of making that not happen, and by embedding it with other kinds of materials or keeping water out from wood. Yeah, you all know about wood swelling and shrinking, untreated wood swelling and shrinking just with humidity changes, right? In the winter, water is driven out of woody surfaces and it'll start to shrink and crack. And in the summer, it absorbs water and starts to swell, and your doors no longer fit into the door jamb so well, perhaps, or there's other kinds of experiences where a swelling of wood occurs. So that's a property that is inherent in these kinds of biomaterials and needs to be managed essentially based upon the type of use.
Great, thank you. Maybe a follow-up question: if you engineer the composition of the cell wall, will, for instance, in wood, will it still be able to be composted?
Yeah, so can it still be composted? So if you use the biological genetic engineering tools that we have in hand, the answer is yes. So we can't, at this point, we aren't able to change things so drastically that biological organisms would no longer be able to break it down. So composting is you keep the material wet, right? The importance of wet. Water is important, and then fungi and bacteria can grow in the stuff and break it down. And there's some materials that are more resistant to that process than others. And so the compost changes its properties with time, and eventually in nature this becomes the organic humic material. So these are residues of lignin and suberin, which are polymers that are made by plants largely to resist water and to and they're good resistances to bacterial and enzymatic attacks, as well as breaking down in your compost. It slows down the compost, and of course that's good because you wouldn't want your compost pile to totally go up into the atmosphere, right? If the microorganisms digested everything, you wouldn't have anything to put into your garden. I'm assuming you're composting at least in part for gardeners. So yes, so the answer is within the purview that we can have right now, it would all be compostable. Even strange biopolymers that are made by novel biochemical pathways, even those are all highly digestible by microorganisms.
Great, thank you. Maybe on sort of a related note, Lynn Heritage asks a question: please explain how the cellulose of firewood burns and releases heat.
So how does it release heat? So it's an oxidation where oxygen is combined with the carbons, and in doing so, it changes the bonding arrangements, and this is an exergonic process, at least in our atmosphere, right? So in other atmospheres, it might be a very different chemical process. And so when wood is heated up, what happens is the things like cellulose and the other polymers start to break down thermally, and they start to vaporize. And as they vaporize, they're broken down thermally into smaller molecules that then are volatile, and then they can interact at this high temperature with oxygen and as a consequence, energy is released. Our bodies do the same sort of thing. They take starch, they break it down enzymatically into glucose units, and then that glucose is burned, essentially combined with oxygen, but not through a thermal, purely chemical process, but under the control of enzymes that will stepwise take it into smaller and smaller units, and then in the final piece will combine it with oxygen, and heat is generated, but in the process, some of that energy is harvested to drive into other chemical forms that are then used to drive our metabolism. Right?
Great, thank you. Yeah, I like that analogy between, burning wood and our own body burning calories to generate, we generate some heat, but also generate energy for our own use. So the patron of this lecture series, Abai Ashtakar, asks, what is the time scale envisioned for wood to replace steel in building of skyscrapers?
So my understanding is that this is ongoing right now. So people are actually processing wood, making it into these very strong laminates that are strong enough and have the proper physical properties to replace steel. And there are a number of, across the world, a number of building designers that are driven by, at this point, probably eco-consciousness, to build these high rises. I'm not sure they quite yet reach the scale of skyscrapers, New York style skyscrapers, but much larger than the 2-story, 3-story buildings that we live in. And those are happening right now. I haven't looked into it, but 2 or 3 years ago there were a couple in process. So I imagine they're continuing to build. And there's more ambitions in that direction.
Great, thank you. Yeah, I would point out Penn State's own ecosystem science and management building, which contains a lot of the forestry research on campus, has a lot of structural wood and also some really beautiful sculptural wood in that building. So if people are visiting Penn State in the future and want to check that out, that's a good showcase of different wood building techniques. So the next question is from John Stitzinger, who asks, will we finally see commercial transparent wood? He notes that it's been around since 1992, but is now in the news this month.
Yeah, will we see transparent wood commercially? So there are materials. There's I'm not in touch with that field to really give an authoritative answer. So I don't know when that will actually show up as products. It'll probably show up in novelty products, I imagine, and then ultimately into something that really finds a strong commercial usage. But I'm sorry to say I don't really have an authoritative answer on that. I'd have to Google it to find out. Right.
Okay, great. Another attendee asks, while they understand the value of research simply for its own sake and don't wish to diminish that by asking this question, so you mentioned wood, paper, cotton, etc. But could you give any examples of how this research significantly impacts some day-to-day experience in people's lives?
So the research I'm talking about is fundamental science. All right, it's funded by the Office of Science in the Department of Energy, and science is the first stage for technology. So what you're asking is, how is the science being converted into technologies that then are used to benefit humanity? At this stage, we are doing on this basic platform of basic science, understanding how it works. Right? That's our remit. That's our goal. Is there a technology that has been? Is there a material or process that has been directly modified by our center result? I don't think so. We're still early in the game.
Great, thank
I
you.
would also point out that there is a division of labor here a bit. So scientists are scientists. They investigate the way the universe works. That's our object. Engineers are engineers. They try to take the basic scientific principles and convert it into things that modify. We do have some engineers in our center, and so we are crossing over into that border in some way. But it's still very science-oriented research.
Right. Yeah, I can speak a little bit from my own experience in the center. And one of the great things about Penn State is that we collaborate quite well across disciplines. So I've had discussions during my experience in the center, talking with engineers, for instance, who are trying to make nano patterned materials. And Dr. Cosgrove mentioned the fact that cellulose and these other components of the cell wall make these very specific nanoscale patterns. And we had talked about using that nanoscale patterning to actually then fabricate metals and other objects that have those specific nanoscale patterns. And those could be useful, for instance, for making catalysts or other materials that would then be put into industrial use. So that's just one example of kind of this crossover between our understanding of plant cell wall structure and potential engineering at that tiny scale. So we have another question from an attendee, and they're wondering what gives bamboo its strength and flexibility, and does it have a special cell wall structure?
So bamboo, the organization, the anatomical organization of bamboo is rather different than wood, for example. So if you do a cross section of wood, you see these bundles of fiber cells now that run up through the body of the plant, through the stem, and that is that organization, and those bundles are embedded in a much softer matrix, so softer cell walls that are more flexible. So those bundles then are inherently more flexible, the way thread, for example, is a highly flexible object, and yet it still has mechanical strength if you try pulling on it, for example. It still then then the tensile properties of cellulose, which are tremendous. One of the reasons people are interested in nanocellulosics, for example, as a way of taking cellulose fibers, which have this long length but very small diameter, embedding them in other materials, perhaps in an organized way. So plant cells do this every day, right? They take the cellulose microfibril and put it out into the cell wall, embed it in an object, and they can make things that are very strong and flexible, or much more compression resistant, by arranging these polymers. So ultimately, I think bamboo has those properties because of the way the plant cells organize their fiber cells, which are cellulose-rich materials.
Great, thank you. Yeah, I'm always struck by the strength and grace of bamboo. There's a great little bamboo grove as part of Penn State's Arboretum that it's a lot of fun to walk through and contemplate that, and how fast it grows too.
There's a great article in National Geographic maybe a dozen years ago on the wonderful properties of bamboo, and I encourage the audience to Google it and find that article. It's quite informative and interesting.
Great. Yeah, one of the other center members, Jim Kubicki, notes that there's high amounts of silica in bamboo as well, and that's one other difference between many types of wood. Great, thank you. One other point I would make on getting back to the question about how is this research potentially applicable to daily life. One of the researchers who was formerly supported by the Center for Lignocellulose Structure and Formation, Jeff Catchmark, has developed a material that's a combination of a modified form of cellulose called carboxymethylcellulose and chitin. And chitin is material that you can get, for instance, from the waste products of shrimp and lobsters and crabs. So by putting those two materials together, he was able to develop a biologically based plastic analog that could be used for food coatings and potentially replace some of the less degradable food coatings that are currently used in the food industry. So if you have an interest in that, I encourage you to Google Jeffrey Catchmark and cellulose and chitin, and you can find out a little bit more about his research there.
Thanks, Jeff. That's a valuable comment, and that opens up the topic of cellulose-based films or biological polymer-based films. And there's a huge interest in this by biopolymer chemists to make films from cellulose and other polysaccharides in the cell wall, as well as Jeff mentioned, polysaccharides from other organisms, chitin being an example of that. If you look at their structures, you find them, they're all based upon sugars and slight sugar derivatives that are organized into chains, sometimes long linear chains, sometimes branch chains. Those are synthesized by these by organisms, and then they can be extracted, purified, put together in novel combinations to make films, which have to that need to have special properties. So that, for example, they have to resist or allow exchange of gases. That's an important thing for a food packaging material. You don't want it to allow too much water to get out, so it dehydrates, but you also have to worry about trapping some kinds of gases inside this living food object. And so there's a tremendous industry built upon that, and a renewed interest these days, because these films, biofilms, are these biopolymer-based films are intended to replace these plastic films that are used currently, which are much less degradable and also are based upon petroleum. And if we want to, and the world wants to get away from building our world upon the petroleum industry.
Right.
With apology to the petroleum engineers who are listening in.
Sure. Yeah, it's amazing to think about how, those towering redwoods that you showed at the beginning of your talk are composed largely of sugar, right? That they're made of these sugar-based polymers and have been able to spin those into, this massively strong and amazing structure. So great.
Yeah, 300 feet high.
Yeah. All right, well, that concludes our question and answer period. When you exit today's lecture, you'll be given the opportunity to provide feedback about today's presentation. Your feedback helps us ensure that these lectures are meeting our objectives and your informational needs. I want to thank everyone for attending this fourth presentation in the Ashtekar Frontiers of Science lectures in the Eberly College of Science. Our next lecture will be on Saturday, February 20th, using the same Zoom link, when Dr. Ileana Baums, professor in the Department of Biology at Penn State, will present a talk entitled How Coral May Survive Climate Change. Dr. Cosgrove, thank you again, and I wish everyone a good week.
Thanks, and goodbye, everyone.